My name's Vincent Riggi, and as the slide says, I'm going to be talking about investigation of individual and combined effects of blah, 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 blah. That's boring, so we're going to get rid of it. I think a better title for this talk would be What Works and What Works on Earth. And I'm going to be talking specifically about RNA oligomerization, but this can really be applied to any sort of biological or prebiotic product that we're interested in. So I want to start out by talking about two different approaches to prebiotic chemical experiments. The first of which is a more traditional approach, and we start out by looking at modern life. We take these cells, we look at what's inside of them, we break them down into these simpler components. We sort of pick our favorite component and then try to replicate it in the lab, abiotically. Then we try to take the parameters that work and apply them to the early Earth. So we look at this and we say, where could this have happened? But the problem is, more often than not, we get a system that doesn't look like anywhere on the early Earth, or very dissimilar to the early Earth. So an alternative approach would be to start with the Earth. We can take the Earth and take different parameters that describe the different environments that we think were around before life, and replicate those conditions in the lab, and see what comes out of it. So as I said, I'm going to be talking about RNA here, but this can be proteins or amino acids or whatever you like. So we have conditions that foster prebiotic chemical reactions, and we have conditions that were present on the early Earth. The key to finding the pathways that are feasible for prebiotic chemistry is to look in the overlap between these two sets of conditions. So if we're applying this to RNA oligomerization, we might be interested in several variables, including temperature, pressure, and any catalysts. And for the sake of this talk, we're going to stick with these three, and we're going to be talking about aqueous catalysts. So this figure shows all of the conditions, at least prior to this week, that have been explored in a laboratory setting relating to RNA polymerization. And we can compare that to the PT space that we think represents early Earth surface environments. And we see immediately that the majority of this PT space has not been filled out yet. We're sort of stuck at 1 bar and sort of moderate temperatures. We did a decent job with temperatures as a community. And we could do a similar comparison for our catalysts. So we have several sets of metal catalysts, including uranium and the lanthanides, that have been investigated as effective catalysts for oligomerization. And some of these work and some of them don't work so well. But we can compare that to the elements that we would expect to find in a prebiotic early Earth environment, such as the open ocean. So in that case, the major cations would be sodium, magnesium, calcium, and iron. So there's not a lot of overlap, but there is some overlap. We have magnesium and calcium. We're still missing sodium and iron. But let's take magnesium and calcium for a second. Those have only been explored at one specific concentration by the Sawai group in the 70s and 80s. And that was only at 12.5 millimolar. So we don't know what increasing or decreasing the concentration would really do. So we have this large empty plot with one data point on it. So we don't know what the rest of this looks like. So what do we do? Well, we do experiments in our lab. These are relatively simple experiments. They're done in microcentrifuge tubes. They're 100 microliter experiments. And all we do is add our aqueous metal solution at the concentration that we're interested in. And we add our nucleotide. Now, in this case, we're using a phosphoramidazolide. This is an activated nucleotide. It's not particularly realistic in its own right, necessarily. But it is useful in the lab for probing these reactions and seeing how they'll behave and respond to different variables like temperature, pressure, and different catalysts. So we take that solution, we wait 3 days, and then we extract our products and we measure them with MALDI-TOF MS. And we get a mass spec that looks something like this, where each peak corresponds to the addition of a single nucleotide. And again, this could be a polypeptide or whatever you're studying. So here we have, oops. Here we have a max length of 6. So this is how we're going to denote the success or assess the extent of this reaction going forward. So you got a preview of these results. But these are those max lengths for different concentrations of calcium chloride. And we see that there's a sort of optimal concentration, right around 1 molar. But this is a relatively broad profile here, and it works over a range of concentrations. We can also compare that to our negative controls, which are represented by the orange line. This is containing no metal catalyst. It's just water and IMPE. But again, we don't want to be stuck at one PT point. We want to expand this and look at a range of temperatures and also a range of pressures. And if we do experiments at 1 kilobar, for example, perhaps we can then extrapolate and then fill out a large chunk of this PT space. We can do our high pressure experiments in a static pressure vessel by loading our experiments into syringes. And these syringes contain our experiments, and when we apply pressure with water, we use a screw pump. That pressure is redirected to the sample. So we have our concentration profile. We need to pick 1 or 2 or however many concentration points that we're interested in to do our other experiments. So we're going to pick 1 molar. And this is what we get. We have our 1 bar, our atmospheric experiments, in orange, and our 1 kilobar experiments in white. And we see that there really isn't a large difference between these two. So we can say, in this particular system, not all systems, that pressure isn't particularly significant. We also see that there's a moderate decrease in oligomer length as you increase temperature. So we can do this with the other metals as well. We have calcium. We did this with sodium and magnesium. But what about iron? We've heard a lot about iron today and throughout the conference. What can it do for RNA? We know it's good for amino acid synthesis and a variety of other reactions. What about RNA oligomerization? But this is a little bit more complicated because we have to keep our systems anoxic, otherwise we'll just end up with a rusty tube. So instead of using a microcentrifuge tube, we use gas-tight excitainer vials. We load our nucleotide into these vials, and we flush the headspace with pure nitrogen to expel any oxygen. Then we inject our anoxic iron solution. Again, we wait 3 days, extract our products, and we end up with mass spec. So these are the results of our iron experiments. And again, well, note first that we're on a log scale now, just to make everything easier to see. But again, we end up with a local optimum concentration right around 100 millimolar. And again, we have our negative control down here at 2. But we get up to about 6. We don't want to be stuck, so we explored this optimal concentration over temperature as well, just like we did for calcium. And we get a nice linear relationship with temperature. But this time, all of these experiments are above our baseline, our negative control here. So even at our highest temperatures, we're still seeing an enhancement of oligomerization. However, this concentration is outside the feasible limits for prebiotic iron in the early ocean. So we have our optimal concentration, and then this is sort of the range of estimates in the literature for prebiotic ocean iron. So we have this other point up here. This is sort of the maximum feasible concentration. So let's take that instead and ask ourselves the same question. Well, this is what it looks like in blue. And we don't see that decrease in oligomerization with temperature. In fact, we beat out the higher concentration when we get out to 80 or 90. So we've added some of these missing elements to our table. We have our 4 primary metal cations in the early ocean. What can we do with that? Well, we can start to ask ourselves what a more complex environment might look like. But we're still going to keep it relatively simple. We're going to look at the open ocean. We have our controls, our negative and our positive control. So 2 for no catalyst, and with 1 molar calcium chloride, which is what we use as our positive, we get pentamers. And then before we put everything in one pot, let's just make sure we know what each thing does individually. So if we look at calcium, at modern ocean concentrations, we see that we get trimers. Sodium sulfate, we get dimers. Magnesium chloride, trimers. And sodium chloride, dimers. So when we put these together, we can ask ourselves, okay, are we going to get trimers, or are we going to get dimers, or are we going to get 3 plus 2 plus 3 plus 2? Turns out that we just get 3, so these aren't additive, at least in terms of the lengths that we get. We can't really say anything about yields because we're using MALDI and it's not quantifiable. But we're interested in the early ocean, so we add in iron. And when we mix that with our other components at our other salts at modern concentrations and take out the sulfate, because there wasn't any sulfate, we can run similar experiments, and we see that, again, the biggest sort of wins. So iron, which is up here at 5, is pretty close to what we get for our iron-rich synthetic seawater. Let's push it a little bit farther, because we don't know exactly what the Hadean looked like, as we've discussed throughout this conference. So let's test a range of salinities and a range of temperatures. So here we have twice modern salinity, half modern salinity, and modern salinity in green. And we see that, as we would probably expect, slightly higher salinity produces slightly longer oligomers. All of these are above our negative control. And as I showed before, there's no relationship with temperature because we're sort of below that 10 millimolar level for iron. So our takeaway points from this is that iron is very significant, not only for RNA synthesis, but for a variety of prebiotic reactions. But more than that, I want to emphasize the importance of starting with environmental considerations and working our way up from there. And it doesn't have to be Earth. It can be Europa or Enceladus or Titan or whatever we're interested in. But we have to interpret our data and design our experiments with our environment in mind. That way we're not shoehorning an environment onto the early Earth. So with that, I'll thank our funding agencies, and I'll take any questions if there's time.